Hi engineers, so what we're going to do in this video is we're going to continue. We left off talking about what happened during the gastrulation process and the development of the notochord. What we're going to do is we're going to take it from that point of where we left off, right, with our notochord, our trilaminar disc, and how we're going to completely develop the nervous system. It's going to be awesome. Let's go ahead and get started. All right, guys, so we ended off last when we talked about the trilaminar disc, right? So we talked about how we formed the ectoderm, mesoderm, endoderm, and then we finished lastly with how what happened is, remember we said that whenever the cells are moving through that primitive pit, right, and we technically can call those cells that are moving through there, well, they actually are kind of like mesodermal derived cells. They form the notochord, right? Now, what happens is, usually to kind of keep up, we were at the end of the second week. If you really want to be particular, we might be close to about day 15, anywhere from day 15 to day 17. It's about where we're at right now. Now, what happens is the body is so cool. That notochord, that good old notochord, it starts releasing certain types of growth factors, right? So this notochord starts secreting a bunch of different types of growth factors. And when it does this, it tells the ectodermal cells start proliferating. So again, first step here, notochord is going to release chemicals. And what's this whole process, this whole first event going to be? It's going to be causing ectodermal proliferation, baby. And that ectodermal proliferation is going to occur and occur and occur, and guess what starts to form in the ectoderm? Okay, so that notochord was releasing a lot of those growth factors, many different types. It might be a part of growth factors like such as fibroblast. It might be transformer growth factor beta. There can also be parts of like the Wnt pathway, the nodal pathway. There's so many different types of pathways that are involved in this. Not necessarily the most crucial thing. What I do want you to know is, as the ectoderm starts proliferating and thickening, it forms a specific type of plate, right, which we call the neural plate. So again, ectodermal proliferation will lead to this kind of like this big thickening of the ectoderm, which is going to be called the neural plate. Now, as we get this thickening of the neural plate, what starts happening is the embryo will actually begin to start allowing for this plate to begin to kind of involute, right? So imagine in like this way. Here's the plate. Imagine I'm pulling in this central line, right? What's dividing in this into two pieces. Imagine I'm tugging on that from the bottom, right in the center. It's going to pull the center of this plate downwards, right? At the same time, the edges of this neural plate are going to start working towards one another. They're going to come towards the center, right? So again, central part of the neural plate, pull that down. Edges of the neural plate start rolling towards one another, right? So that's going to begin this kind of folding or involution process. As that begins to occur, look what we get next. Look at that. So now, remember that part I said was here at the bottom? Imagine here is like that line that we had there, right? And that's spanning all the way down here from the part of where we had that primitive pit, if you guys remember, all the way up to the actual prochordal plate. Now what's happening is, from that part, it's pulling down, it's involuting, and then the edges of that actual plate are starting to move towards one another. Now, what happens is you get this part here. You see how it's kind of like folding? It's getting ready to fold. This is going to be called our neural folds. And then here, this little like U shape or half a circle or almost a complete circle, almost a complete tube, this is actually going to be called the neural groove, right? Now what happens is some cells start to actually specialize and differentiate, right? Some of those ectodermal cells start to proliferate, differentiate, and become specialized cells. You see these orange cells? These orange cells are what we call neural crest cells. And they kind of form towards around the edges of the neural folds. So the neural folds is the edges of the neural plate. The neural groove is going to dip down, make this little kind of like canal. And then the neural crest cells are going to be kind of right around the edges of the neural folds. So again, what are these orange cells here called? They're called neural crest cells. These are important because they're going to differentiate into a lot of different structures that are important for our peripheral nervous system, okay? So again, neural folds, neural groove is going to begin to happen, right? Around what day do we start to see the neural plate, right? So when does this notochord start releasing certain types of cytokines and growth factors that trigger the neural plate? Usually, approximately day 17 is when we start to see it. So you start seeing the formation of the neural plate approximately around day 17, okay? Somewhere around that. Now, neural folds with the neural groove and the neural crest cells, they start kind of growing approximately around day 18, so the next day. So you'll see this around day 18. Now, here's where it gets really good, right? The neural folds, the edges of the neural folds fuse with one another, right? The neural groove, it completely involutes, and then you form a complete tube, right? That tube comes underneath the ectoderm, and then we form this neural tube. Look at that. Look at that little guy right there. That's our neural tube. So right here, we're going to have the neural tube. And remember, what do we say? We had the neural folds, right? So remember, it's kind of coming in. The neural folds are fusing with one another, and then right up on top of them, you have your neural crest cells. Well, look where they're forming. Right there where the neural folds fuse together. So cool, right? So again, that's our neural crest cells still. So neural crest cells. And then again, what's here below it? The notochord, right? So you still have the notochord, and then the ectoderm is going to be there, right? Now, what's important is we need to look at this from two different views. It's nice to look at it like this, but it's also nice to look at it from an actual kind of a larger view, right? So kind of like a more of a dorsal view. So now, let's take, we had this here where the actual neural folds fused, right? You get your complete neural tube with the neural crest cells just above it and the notochord below it. But look at this view. It's the same view. Instead of us looking at it and the tube kind of straight on, now we're looking at kind of a dorsal view, right? Now, or a top-up view. Here is going to be the cranial portion. Okay, so up here, let's mark this in purple. This is the cranial portion of the neural tube. And this is going to be the caudal portion of the neural tube. Now, there's a part here of the neural tube, right, which we call the posterior neuropore. In other words, if you imagine kind of like this, here's a tube, here's the other end of the tube, right? This tube hasn't completely closed off, right? So it's open. It's kind of like a vessel, basically. Let's say that this part up here is the cranial, this part here is the caudal. This is going to be what they call the anterior neuropore, and this one here on the bottom is going to be the posterior neuropore. Now, around approximately we'd say around day 21, you completely form the neural tube. Here's my pink marker, there it is. So around day 21, you completely form the actual neural tube, okay? But the neuropores are still open around day 21, okay? As the cells continue to proliferate, eventually they close up that cranial end and that caudal end, or what we call again, what do we call this area here? Anterior neuropore and posterior neuropore. Why am I mentioning this? Why am I spending so much time on it? The reason why is, in order for this anterior neuropore and posterior neuropore to close, your cells need a specific type of fuel, right? And that is called folate. Folate is so important in order to be able to allow for the cells to synthesize DNA and replicate. So with the lack of folate, what do you think would happen to the anterior neuropore and posterior neuropore? Do you think it'll close? No. If it doesn't close, guess what can happen? You can get defects within the spinal cord. So for example, things that can happen whenever the anterior neuropore doesn't close, you can get things like anencephaly. You can get things like an encephalophyll, right? What about a posterior neuropore? What if that sucker doesn't close? Then you can get spina bifida, which has the different types, right? So you have spina bifida cystica and spina bifida occulta. And then there can be complications or associations with that, such as Arnold-Chiari type 2 malformation, Dandy-Walker syndrome, tethered cord syndrome. So there can be terrible things, even with the anterior neuropore. And we talked about, I said, encephalophyll. That can actually lead to what's called Meckel-Gruber syndrome, which can be terrible, where they have cystic renal dysplasia, they have polydactyly, and they have a part of the brain and meningeal tissue that are coming out in the occipital part, right? So these are very, very important things. And for folate, usually during pregnancy, you need anywhere from about 3 to 5 milligrams per day to be able to maintain this actual proliferative process, okay? So folate is absolutely necessary for the neuropore closure. Okay, around What day approximately do we say the anterior neuropore closes? The anterior neuropore generally closes approximately around day 24. So around day 24, the anterior neuropore closes. And that's a good thing. We don't want that bad boy to be open. Again, if it is, it can lead to anencephaly, it can lead to an encephalocele, right? Around day 26, approximately, the posterior neuropore should close. So that should be the posterior neuropore should close. All right? Now, once the posterior neuropore has closed and the anterior neuropore is closed, then what can happen is we can take this nice little spinal tube, right? Or basically what we're going to lead to the development of the entire central nervous system. So this neural tube that we formed is going to lead to the development of the entire central nervous system. It's unbelievable. So we're going to see how that happens. So to make it simple for you, what I'm going to do is imagine I take here, right? So you're going to have the anterior neuropore, posterior neuropore, cranial or cephalic end, caudal end. All I'm going to do is take this and just imagine you have a simple tube, right? Top part here, bottom part here. Cephalic end here, caudal end here. Cranial end here, caudal end here. It's the same thing for all of them. And see what happens when we take this little tube and we develop all these different structures. It's so darn cool. All right, so again, what have we covered throughout this entire process, all the way from where we started with our trilaminar disc, all the way until we formed our neural tube? That was the process of neurulation. All right, so again, just remember that this is neurulation. Very simple process, nothing crazy to it. It's just basically a step of knowing, right? Ectodermal proliferation, neural plate, neural groove with the neural folds, neural crest cells forming on the edges of those neural folds, completely closing up, forming the neural tube. And then after that, anterior neuropore closure at 24, posterior neuropore closure at 26, which requires folic acid, or folate, around approximately 3 to 5 milligrams. All right, so what we're going to do now is we're going to take this tube that we formed through the process of neurulation, right? And all we're doing is we're kind of just pulling it out of this structure that we formed over there, right? Now, what we need to do is take this neural tube, form the central nervous system. So we have to go through a process called vesiculation. So what happens is this neural tube towards the actual, what is this end? We got to make sure that we know our ends. This end up here is going to be the cranial or cephalic end, whichever one you prefer. It doesn't matter. Caudal end, which is going to be the tail end, right? Towards the cranial end, we're going to form 3 vesicles. It's kind of bulge out. Those 3 vesicles are going to be these from cranial to caudal accordingly. First one is called the prosencephalon. Okay? The second one, which is going to be the actual purple one, is going to be called the mesencephalon. And this is basically going to become the midbrain. The prosencephalon will separate into 2 other vesicles in a second. And the green one here is actually going to be the rhombencephalon. They had some names for these in the past, right? But again, accordingly, rhombencephalon, mesencephalon, and prosencephalon. And again, what are these structures that we form first? This is going to be the primary vesicles. So primary vesicles. Now, what happens is these vesicles, they even develop even more. So they proliferate, they start specializing, they start curving in different ways, and they form some more vesicles. Now, remember we had the prosencephalon? What I want you to remember is that splits into 2 parts, okay? It splits into this top one, right? That kind of looks like your telescopes. This is going to be called the telencephalon. So the telencephalon is this top one. Now the prosencephalon forms the telencephalon, and it forms this kind of like maroonish one, which we call the diencephalon. The diencephalon. And then this purple one, you guys should already remember, we already covered it, it stays the same. It's the mesencephalon. That's going to become the midbrain. So this is still the mesencephalon. cephalon. Then the rhombencephalon is actually going to break up into a couple different pieces, right? All right, so this green structure here is going to be the mesencephalon. All right, now the mesencephalon is going to be basically become the pons. But there's another structure that's a part of the mesencephalon. You see these little like peanuts that are coming off the edge over here? These little guys here are going to be forming a part of, they're still a part of the mesencephalon, but they're going to become a part of the cerebellum. They're going to become the cerebellum. Then you got these orange structures here, right? So then you got this next one. And this is going to be the myelencephalon. And that's going to become the medulla. So prosencephalon becomes the telencephalon, which is going to be your cerebral hemispheres, and the diencephalon, which is going to be your thalamus, your hypothalamus, your epithalamus, your subthalamus, mammillary bodies, all those good jazz. Mesencephalon will stay the same. It's the midbrain. The rhombencephalon will form the mesencephalon, right, which is going to become the pons and the cerebellum, and then the myelencephalon is going to become the medulla. And then guess what the remaining part here is? The spinal cord. And we'll talk about how that develops in a little bit. So again, this last part here is going to become the spinal cord. All right, so to finish it all off now, telencephalon is going to become this structure, right? And this is going to become the cerebrum. So we're going to have the cerebrum, which has your different lobes. You guys know frontal lobe, parietal lobe, occipital lobe, okay? Even the fifth mini lobe, which is the insula, temporal lobe. Then inside of that cerebrum, you're going to have little gray matter structures sprinkled in there. And remember I told you that's a part of the diencephalon. So a couple of those structures are going to be your thalamus, right? You're going to have the subthalamus. What else are you going to have? You also have the hypothalamus. We're not going to show that here. And you'll also have the epithalamus, or the part of the pineal gland, right? So we'll have over here that's going to be a part of the epithalamus or the pineal gland, which is going to also consist of other structures as well. So that's going to be your diencephalon, right? And again, it's going to consist of thalamus, epithalamus, hypothalamus, subthalamus, and even a part of the mammillary bodies. Then you have your mesencephalon. What did I say that's going to become? The midbrain. So midbrain. And then the mesencephalon is going to become two structures, right? We said that that's going to become what two? This green one here is going to become the pons. And then the dark green is going to be the cerebellum. And then we also have the orange here, which is going to be the medulla. Medulla oblongata. And then lastly, again, we'll have the spinal cord. So it's amazing to see how from a little tube you get your primary vesicles, prosencephalon, mesencephalon, rhombencephalon. Prosencephalon makes telencephalon, diencephalon. Mesencephalon stays the same. Rhombencephalon makes the mesencephalon and myelencephalon. Telencephalon is going to form your cerebrum, right? All the different lobes. Diencephalon is going to be the thalamus, subthalamus, epithalamus, mammillary bodies, hypothalamus. Mesencephalon is going to be technically the midbrain. Mesencephalon is going to be the pons and a part of the cerebellum. And then the myelencephalon is the medulla, and then we'll have our spinal cord, and we'll talk about all the structures within that part of the spinal cord. So it's an amazing process that can happen here, which is this process of vesiculation. Okay, so what is this process that we basically covered here, going all the way for the development of the brain stem, the cerebellum, and we'll get into more detail in the spinal cord. This was the process of vesiculation, right? So vesiculation. Now we got to keep going, right? So we took that tube there, right? That neural tube, and we formed these structures. But remember, there were some cells that were right on the kind of surrounding that neural tube. Remember that? We talked about the neural crest cells. These little suckers are so darn cool. So what we have to talk about in this part here is I want us to discuss the neural crest cell differentiation. Because these little buggers, they just do a bunch of things. They turn into a bunch of different structures. So what do these little guys do? Well, they move all over the place. They're just moving, right? And they become multiple different types of structures. Where? All over the place. So what are some of the things that they can become? Well, you know your skin cells, your melanocytes that are within the stratum basale, right, of the skin, the epidermis, they're going to become those. They're going to become the melanocytes. The melanocytes, what are they important for? They produce pigment that allows for us to be white or brown or black, all the different types of colors. So again, they're going to become melanocytes. What else? They can migrate to these little structures that are sitting on top of our kidneys, where they can become what's called the enterochromaffin cells, or just the chromaffin cells, that secrete epinephrine and norepinephrine that are important within the actual catecholamine surge for our stress response. What is this? They can actually come down here and form and become a part of the adrenal medulla. And again, this is a part of the sympathetic nervous system, right? So again, you know the sympathetic nervous system, you have preganglionic axons that come out into this adrenal medulla, and those are going to be long. They come and they synapse on these little postganglionic motor neurons, which are very short, and they release lots of epinephrine and norepinephrine. So again, remember these are important because they make epinephrine, and they also make norepinephrine. And if you remember, it's 80-20. Pretty cool stuff, right? All right, next thing, they also can actually develop not into the actual structures of our gut, right? Because that's the part of the endoderm, but they can derive into the nerves that are important in surrounding that structure. If you remember the submucosal nerve plexus, right? We also call it the Meissner's plexus, or if you guys remember the myenteric plexus or the Arbach plexus, they're important with what structure? The enteric nervous system. And they're all within that submucosa. And what else? Muscularis externa. So what are these structures here? These darn things are important, right? Because they help to involve structures for the enteric nervous system. Enteric nervous system. And again, what are some of the structures of the enteric nervous system in case you might have forgot? Remember, you have the submucosal plexus, and then another one is going to be the myenteric plexus. And if you like the old school name, you can also remember submucosal is also known as Meissner's. Don't know how to spell it, not going to. Myenteric plexus is Arbach's plexus. So that's pretty cool, right? That helps to regulate our GIT. All right, what else? They can also migrate just outside of the spinal cord into these neurons, these pseudo-unipolar neurons that are located outside of the spinal cord. What are these little guys? Well, if you remember, a lot of the sensory information comes into these little pseudo-unipolar neurons, which has a peripheral process picking up information from the periphery, and a central process which is going into the actual spinal cord. That's our dorsal root ganglion. So the dorsal root ganglion should be here. So here's the dorsal root ganglion. So they're important also for making that. But guess what else? Not just that. Let's amp it up. Let's involve the sympathetic ganglia. This is going to be where the what? You know how you have the lateral gray horn, right? The lateral gray horn, which is particularly within T1 to L2, right? Which is for the sympathetic ganglia, the preganglionic. The preganglionic sympathetic fibers are supposed to be short. They're only supposed to go a small distance where they go into these little chain ganglia or the prevertebral, right? So you have paravertebral, right? Or chain, right? And then over here you have prevertebral or collateral. It's whichever one you like. But what's important is that the preganglionic fibers are going into where these cell bodies are, and then those axons move out. Guess what these are? Again, let's write them down. Geez, my marker's all over, but here we go. So this one is actually going to be the chain ganglia. And this one is going to be your collateral ganglia. This is basically all part of the sympathetic nervous system ganglia. Sympathetic chain, paravertebral, prevertebral, collateral ganglia. They also can develop into these. So darn cool. All right, what else? Neurocrest cells can also develop into these layers of the brain. You know, you have the brain, right? And then you have the spinal cord. And then surrounding that, you have the first layer, which is clinging to the brain and the spinal cord. That little pink layer, what is that guy called? That's called the pia mater. So this is the pia mater. And then what's just outside of that, which is going to be forming this spider-like layer, where the subarachnoid space is, where there's lots of cerebrospinal fluid. This is the arachnoid. mater. Now together, these two maters make up what's called the leptomeninges. Okay? So the leptomeninges are also going to be made by the neural crest cells. Pretty darn cool, right? Now, the neural crest cells also help with formation of a lot of the muscles and bones and different types of structures that form the head and the neck, right? As well as the nerves that are going to be supplying parts of those pharyngeal arches, right? So you have pharyngeal arches. We're not going to go into that right now. In another video, we will. But the head and the neck, and then you're going to have nerves which are supplying the pharyngeal arches, right? What are some of the nerves that supply the pharyngeal arches? Well, the first pharyngeal arch, you should remember, is going to be cranial nerve 5, the trigeminal nerve. So that's going to be one. Neural crest cells help to be able to form those. Also, cranial nerve 7, which is the facial nerve. That's going to be a part of the actual pharyngeal arches. And 9 and 10. Some books will also say that 3 and 8, some of 3 and 8 can also be a part of what the neural crest cells can differentiate. But for right now, we're just going to say 5, 7, 9, and 10. These are helpful because these are going to be nerves of the pharyngeal arches. All right, now, not only does it help for the formation of 5, 7, 9, and 10 cranial nerves, but also, again, for the structures of the head and neck. So let's also remember structures of the head and neck, muscles, the bones, different things like that. It's also important for. Okay? And if you really want to remember something else, you get little bones in the ear, right? The little bones in the middle ear, the ossicles, right? Malleus incus stapes. It also can develop into those structures too if you really want to remember that. So that's also pretty cool. So again, this is going to be the neural crest cell differentiation. So we covered what the top cranial part of the neural tube forms. We covered what the neural crest cell becomes. Now we got to cover what the bottom part of the actual neural tube develops in. Again, remember what we said here? Don't matter how you remember, just remember your orientation, cranial or cephalic. I like cranial because it's easier to spell, and caudal. Okay, head, tail. We covered this part, we covered the neural crest cells, now let's cover the caudal portion. What I want to do though, to make it easier for ourselves, is to see how it actually derives from the inside out. So what I'm going to do now is I'm going to take a cross section. So what I'm going to do is I'm going to go right there, because generally all of the upper structures, right, brain, brainstem, that's all going to be formed up here. I want to see how the spinal cord develops down here. So I'm going to take a cross section and look on it from the top, okay? Now, we haven't formed the spinal cord yet. What we do have is the neural tube, and I want to look at the neural tube. So imagine I'm cutting it and I'm holding it like this now, and I'm looking down the neural tube. Well, you know the neural tube, it might just look like this thin thing, but it's actually made up of 3 layers, okay? We're not going to go into crazy detail on everything in these layers, but what I want you to know is that these 3 layers are going to be consisting of the green layer, the outermost layer, which is called the marginal zone. This is generally where the neurons are going to start actually forming. So the neurons and their axons. Right here is going to be the mantle zone. And the mantle zone is going to be where you have your neuroblasts, okay? And then inside here is the most inner layer, and this is going to be the neuroepithelial layer. Neuroepithelial zone. And what's happening is these cells are proliferating and they're becoming neuroblasts, and then these neuroblasts are becoming neurons, and they're starting to develop and move their way outward from this central neural tube. So what I wanted to do now is we take this whole tube like we had here and just imagine it as this little circle again. Okay, so imagine this circle contains all of these layers: the marginal zone, the mantle zone, and the neuroepithelial zone. And as it's replicating, right, going from the neuroepithelial zone to the mantle zone to the marginal zone, it's making structures that are going to be extending out from that point, right? What are those things? Well, the first thing that's going to happen here is it's going to make like these little bunny ears that come out here, right? It's going to make these little bunny ears that extend out here. So the cells, again, from the neuroepithelial zone, you're going to get, again, the mantle, marginal, and the cells are going to start working their way out and making these bunny ears that are extending towards the actual posterior part. So if you imagine the spinal cord, if I'm taking a cross section, this is the dorsal surface or the posterior surface, and this is going to be the ventral or the anterior surface of that spinal cord, right? Now, what happens is this neural tube forms structures that are going to be working its way posterior towards the dorsum, and then it's going to have some other structures that it's actually going to work its way. Let's do this color. That's going to work its way anteriorly. Look at this. And these two things are specific structures. They have specific names, right? So here, the top part, the little ears, I want you to remember like the little bunny ears. These are going to be called this is your alar plate. Okay, so that's your alar plate. Your alar plate is going to be forming these little bunny ears that are extending their way dorsally from the actual neural tube. And it's going to be, imagine you have a point of symmetry here. So imagine here we have a sulcus and then a fissure, right? So imagine here you're going to have a point of symmetry, bilateral symmetry. From here, working its way out, like posteriorly and laterally, you're going to form these alar plates. And then ventrally and laterally, you're going to form your basal plates. Why is this important? Because what eventually happens is your alar plate, it's going to start looking like something very, very similar. What is this thing here? Well, if you guys remember, this is going to be called the posterior gray horn. So all this is the posterior gray horn, extending from that neural tube. Then, extending ventrally and laterally is going to be the anterior gray horn. Now, alar plate, right, we said this is going to make, again, this is going to be our ventral gray horn. And then the alar plate is going to make the posterior or dorsal gray horn. What kind of neurons are the posterior gray horn? Are they sensory or are they motor? They're sensory. So these are going to be where your sensory neurons are. And what kind of neurons are going to be forming here in the ventral gray horn or the anterior gray horn? These are going to be motor. And then again, guess what the remnants of that actual neural tube is leaving? Nice little spinal canal. And what's in that spinal canal? Nice little cerebrospinal fluid. So inside of this, you're going to have cerebrospinal fluid. Now, that should cover everything that you need to know for the development of the neural tube and the neural crest cells. All right, engineers, so in this video, we covered a decent amount of information. I hope it made sense. I hope you guys really did enjoy it. I really hope it helped. That's the whole point. That's what we want to make happen, is for you guys to be able to understand this stuff. I know it can be complicated, but I hope we were able to help. We're going to continue this embryology series, and what we're going to do next is we're going to move into the next process, which is taking that cytotrophoblast and syncytiotrophoblast and seeing how it implants and makes the placenta. That'll be our next process that we're going to attack. But I hope this video made sense. I hope you guys did enjoy it. If you guys did, please hit that like button, comment down in the comment section, and please subscribe. Also, if you guys could go down to the description box, go check out our Facebook, our Instagram, our Patreon account, and also our GoFundMe page. If you guys could donate any money, we would truly appreciate it. It would help us to continue to make videos for you guys' enjoyment. But love you guys, Ninja Nerds. You guys are the best fans ever. And as always, until next time.